Okay, uh, this is double education. Those who are presenting different educational programs and motivational speeches for general biology, for freshmen, inclusive educations. Kun dambalii chaanaalii barnoota dabbala educationidha. Akkuma yaadatamu immoo sagantaalee barnootaa fi haasawwannachiisoo gosa barnoota bioloojii kutaa 9 hanga 12 fi akkasuma general biology freshman ammallee inclusive education for freshman kan jedhu dhiyeessuu jalqabuun isaanii yaadatama amma. Akkuma yaadatamu immoo akkuma beekamu chaanaaliin kun kan keessanii namoonni chaanaalii kanaaf haaree taatan like subscribe godhuu kan daganne maaliif wanti isin irraa eegamu kanuma qofaa. Inni bira tooppikii irra qabadhe tooppikii baay'ee bareedaa baay'ee namatti tolu very smart topics. General biology for freshman unit 4 lesson about respirations cellular respirations. Kanaafuu waa'ee cellular respiration qabadeen dhufaa jira waan ta'eef tooppikiin kun barattoota kutaa 11 if baay'ee murteessaadha. Unit 5 metabolism baay'ee adda baasata waan ta'eef even barattoonni kutaa 11 osoo kana hordoftan baay'ee namatti tola. This topic is very important for grade 11 students on unit 5 and also important for general biology for freshman students because if you are understanding it also listening this video carefully, I hope you got excellent A+ grades. I hope because it's very smart way of presenting and also about cellular respiration. Today I'm going to present about bioenergetics and the biosynthesis about cellular respirations. Under this, we are going to talk about cellular respiration in details, glycolysis, about anaerobic respirations, formation of acetyl-CoA or link reaction between glycolysis and citric acid cycles, and also electron transportation about aerobic respirations. You know the difference between aerobic respiration and anaerobic respirations. Garaagarummaa anaerobic respiration fi aerobic respiration sirriitti adda baasanii akka beektan hin beekaa jiru. This anaerobic respiration is a way of respiration. It's carried out in the absence of oxygen. While the tricarboxylic acid cycle and electron transport chain is aerobic respiration because it is carried out respiration in the presence of oxygen. Sorry, aerobic respiration is carried out in the presence of oxygen. So, ladies and gentlemen, the follower of channel education, especially general biology for common course of freshmen, especially natural science students, please, if you are new for my channels, like, subscribe, and share for your friends. If you are our clients or member of our channels, please attend me, and also, if you like my video, make like and share for your friends. This is Double Education channel of education for Ethiopian students, for grade 9, for grade 11s, grade 12s, and also for general freshman students. Because those who are going to take national entrance exams, if you use these materials, I hope you got the question which come from this parts. Okay, let me start one by one. Cellular respirations. Cellular respiration. As you know, cellular respiration is the process in which breakdown of organic compounds such as glucose to produce energy. So, cellular respiration is the process where our bodies break down organic materials and they produce energy for our bodies. It is transformation of chemical energy in food into chemical energies. The chemical energy in food that prepare the way we are using for our bodies. Cellular respiration jechuun adeemsa qaamni keenya organic molecule caccabsee kanneen akka glucose irraa energy itti oomishudha jechuudha. It is a way process of transportation of chemical energy into food in the forms of ATP or adenosine triphosphate. Adeemsa qaamni keenya organic molecule caccabsee gara ATP yookaan currency energy for our cells. Energy qaama keenyaaf barbaachisu kan jijjiiru cellular respiration jennaan jechuudha. ATP is the basic currency of cells. The basic currency of cells. ATP energy qaama keenyaaf yeroo sanitti barbaachistu keessaa isa tokkodha jechuudha. We use this ATP for different purpose. Cell uses ATP to power virtual of their activities. Our cells use ATP for different purpose. For example, to grow, to divide it or to the replications, to replace worn out cell parts or dead parts of cells, execute many other tasks in our cells. This is a chemical structure of ATP. It have 3 phosphate groups, ribose sugars, and also adenine. When our body is using this ATP, the bond of this first phosphate is broken and it's converted into adenosine diphosphate. Kanaafuu qaamni keenya generally ATP wantoota garagaraatiif itti fayyadama. To grow guddinaaf, heddummachuudhaaf, seelilee du'an bakka bu'uudhaaf, yookaan wantoota caasaalee qaama keenyaa ittiin ijaaruuf fayyadamna jechuudha. Caasaan ilaaltuun kun caasaa ATP yoo ta'u, this is used by breakdowning of the third phosphate groups. Okay. How do we get energy from ATP? How we get energy from ATP? Of course, we get energy from ATP by breaking the high energy bonds between last two phosphate in ATP. We get energy from ATP when we are breakdowning of two phosphate, the bond between two phosphate group in ATP, then we are converting into ADP. That process is called hydrolysis, which means breakdowning of that bonds and releasing ADP plus water molecules. Adeemsi nuti ATP irraa energy itti argannu bondii phosphate lamaan jidduu jiru yeroo cabsinu energy arganna. Adeemsi sun immoo hydrolysis jedhama. Hydrolysis jechuun immoo adeemsa bishaan itti dabalee ATP plus water molecules it release ADP. So this is the structure of ATP, how we are getting energy from ATP. This is ATP, which have 3 phosphate groups, but the bond between these 2 phosphate groups is broken down by adding water molecules or by process of hydrolysis. Then we get adenosine diphosphate. Previous, this is triphosphate, which means 3 phosphate. Now we have 2 phosphates, but 1 phosphate is breakdown and we get energy for our cells. This is how we get energy from ATP. So when we come to how do cellular respiration is generate energy from glucose, or how our bodies generate energy from glucose molecules or organic molecules, by the process of cellular respiration, but cellular respiration does not oxidize glucose in cellular respiration does not oxidize glucose in a single explosive step. It is oxidized glucose in many serious pathway of or step of reactions. Rather, we have glucose and other organic fuels are brought down into a series of steps. You have during a series of steps and also catalyzed by many enzymes. This is a cellular reaction is have a series of steps and also catalyzed by different enzymes. At a K steps, electrons are stripped from glucose as hydrogen atoms. The hydrogen have a great role in this cellular respiration because electron is stripped from or moved from glucose as hydrogen atom or during oxidation reaction. So this H electron is travel with proton or hydrogen that as hydrogen atoms. This carries our electrons. The hydrogen atom is not transferred directly to oxygen, or that this hydrogen atom is during cellular respiration is not transferred directly to oxygen. Instead, usually they have a carrier or transport material that's called coenzymes, such as NAD and the fat in cellular respiration. You see during the reaction of aerobic cellular respiration. What is NAD? Or what is fat in cellular respiration? The importance of NAD in cellular respirations. So NAD means the abbreviation of nicotinamide adenine dinucleotides. So NAD is nicotinamide adenine dinucleotides, which means it is used as a coenzyme, and also it is an electron carrier in a redox reactions. NAD is a molecules which is used as a coenzymes and also electron carrier in redox reactions. I hope you know coenzymes. The non-protein parts of enzymes called coenzymes, especially organic or vitamins. Function as oxidizing agent in several of redox step during breakdown of sugars. So the importance of nicotinamide adenine dinucleotides is used as electron carriers and also as oxidizing agent in redox reaction of cellular respirations. NADs accept electron from organic molecule during redox reactions. Then it becomes reduced or we use it as oxidizing agents. During reactions, it accepts electron from organic molecules or from glucose. Then it becomes reduced and it becomes NADH. It get electron and it becomes reduced NADH. This dehydrogenase enzymes remove a pair of hydrogen atoms during oxidizing aids or during redox reaction, and also this dehydrogenase removes this hydrogen from two electrons and two protons from these NADs. The enzymes deliver these two electrons and one proton to this coenzyme NAD so that the NADs become reduced to NADH. The enzymes give protons or hydrogen for NADHs, dehydrogenase enzymes. The other proton is released as hydrogen ion into the surrounding solution during the reactions. Let me see by diagrams. We have foods or glucose. That glucose is released two hydrogens. From these two hydrogens, two electron and two proton or hydrogen is present here. Then one hydrogen and two electron is given to NAD by dehydrogenase enzymes, by this enzyme. That reaction is called oxidation of NAD. Then we get NADH or nicotinamide by reduced forms. Then one hydrogen is released in these forms. This is for NAD, and also this is for surrounding solutions. This is about the releasing of or reduced of NAD. This NAD is a common electron carrier in a cell. So it is common in cell electron carrier. But not only one. There is another electron carrier in a cells. We have our cell keenya keessatti we have another electron carriers kan biraa qabna jechuudha. Flavin adenine dinucleotide illee qabna. It is also used as a coenzyme which accept electron during glucose metabolism and it is reduced to fat H2. There is a difference between NAD and the fat. When NAD is reduced, it becomes NADH, but fat is get two hydrogen atoms. Fat can carry two hydrogen atoms, but NAD can carry one hydrogen atoms. The difference between. But each NADH and the FADH molecules formed during respiration represented stored energy that can be appointed to make ATP. One electron complete fall down and energy gradient from NAD and the FADH to oxygens. Then they carry hydrogen too in mitochondria. Then this energy is our proton is fell down into by gradient into ATP synthesis. Then this hydrogen is converted into ATP. Kanaafuu NADH fi FADH erga hydrogen kana baadhatanii deeman booda immoo yeroo electron transport chain keessa dhufan hydrogen kun irraa release ta'eeti gara ATP synthesis or inner mitochondria erga seenee booda hydrogen kun garamitti jijjiirama gara ATP tti jijjiirama jechuudha. NADin tokko ATP 3 oomisha, FADin tokko immoo ATP 2 oomisha jechuudha. One NADH is produced 3 ATP and one FADH is produced 2 ATP. That's a difference. So NADH and the FADH2 is an important energy intermediary in cells. They also store energy in the form of electrons that can be appointed to make ATP. They have electrons that are appointed to make ATP. They have a lot of stored energy in our cells. We have ATP synthesis in inner membrane of our mitochondria, which is used for ion gradient to power ATP synthesis. This ATP synthesis is one of the most important enzymes, which is take hydrogen from NAD and FADH and they produce ATP by using a ion gradient power to power ATP synthesis. For every hydrogen ion that flows through ATP synthesis, one ATP can be formed from ADP, adenosine diphosphate plus phosphate, it produces ADP. In our cells, we have two ways to make ATP via substrate level phosphorylation and oxidative phosphorylation. Let me say the difference between substrate level phosphorylation and oxidative phosphorylation. Substrate level phosphorylation means it produces a smaller amount of ATP, which means almost it produces 2 ATP, 4 ATP. In this synthesis, ATP is produced when enzyme is transfer a phosphate from substrate to ADP. The source of phosphate for ADP is substrate. Sababa kanaaf karaalee lama seelii keenyaaf ATP ni qophaa'a. Karaan inni jalqabaa substrate level phosphorylation isa jennuuni. Karaan inni lammaffaa immoo oxidative level phosphorylation isa jennuuni. Substrate level phosphorylation jechuun adeemsa ADP'n enzymiin phosphate substrate irraa fuudhee ADP kennee ATP'n itti oomishamu jechuudha. The process where enzymes transfer phosphate from substrate to ADP. ADP phosphate is from substrates, from glucose. When we come to oxidative phosphorylations, it is use of ATP synthesis. It uses enzymes called ATP synthase and the energy derived from protons gradient to make ATP. Proton gradient kan reefu jennaan sana jechuudha. That is carried by NADH and fadiidhaan kan geejjibamu sanirraa ATP oomishaa jechuudha. It produces ATP from proton gradient to make ATP. That is come from NADH and FADH. You see the difference between substrate level phosphorylation and the oxidative level phosphorylation. I hope you understand the difference between these two way of ATP synthesis. But this oxidative phosphorylation is very important than substrate level phosphorylation. Oxidative phosphorylation in kun substrate level phosphorylation irra baay'ee important because percentage 90 ATP ne isa keessatti oomishama oxidative level phosphorylation keessatti oomishama. 90% of ATP is generated by oxidative phosphorylation in respirations. Account for 90% of ATP is generated by respirations. Let me see the difference between glucose and the glycogens. You know what is glycogen, what is glucose. So this glucose is a key metabolite in metabolism, a key nutrient in metabolisms. It is various pathways that is concerned with utilizations, storage and regeneration of glucose exists. Karaalee adda addaatu jira kan ittiin glucose utilize goonu, storage itti goonuu fi regenerate lammaffaatti oomishnu karaalee adda addaatu jira jechuudha. We have different pathways that is utilizations, storage and regeneration of glucose exists. From that ways we have glycogens, a way we are storing glucose in the forms of glycogens. It is a polymeric storage form of glucose in humans. When we have excess glucose in our cells, we have converted in the forms of glycogen. So it is called animal starch. It is most abundant in the livers and also muscle cells and also brain cells. This glycogen is abundant in liver and muscle cells. Qaama keenya keessatti glucose yoo baay'atu hamma glucose dhiiga keenya keessa jiru tasgabbeessuudhaaf qaamni keenya glucose kana garamitti jijjiira, it is converted into glycogens. It is synthesized when glucose supply is high. Hammi glucose qaama keenya keessa jiru yoo high ta'e, we synthesize glycogen. It is degraded help to maintain blood glucose level when we are fasting. When we are in fasting or when we are in lack of glucose. When glycogen is depleted or converting glycogen into glucose by the process of gluconeogenesis. When our bodies or our blood is face lack of glucose during fasting or due to several reasons, our body is converting glycogen. Then the glycogen is become depleted. You see, when our glucose is excess in our blood, we convert it into glycogen. When our body is face lack of glucose, we convert or deplete these glycogens into gluconeogenesis. The process where glycogen is converted into glucose is called gluconeogenesis. Gluconeogenesis. These pathways, most important substrate are amino acids. The pathway is amino acid, which are obtained either from protein-rich diet or during starvation from breakdowning of cellular protein, mainly in skeletal muscles or in muscles. So gluconeogenesis occurs in liver and in kidney. Most of the times it's found in kidney and in livers. So the process we converting this glycogen into glucose is gluconeogenesis. Well, we have two forms of cellular respiration in our bodies. Akkuma beekamu karaalee cellular respiration itti gaggeeffamu lamatu jira. The first one is aerobic respirations and the second one is anaerobic respirations. As we know, as you remember in previous class or in previous experience, akkuma yaadattu muuxannoo keessan yookaan barumsa kana dura barattan waa'ee biology irratti akkuma yaadattan karaalee lamatu jira. The first one is aerobic respiration. The second one is anaerobic respiration. Aerobic respiration means the process of cellular respiration is carried out in the presence of oxygen. Aerobic respiration jechuun cellular respiration bakka oxygen jirutti yeroo gaggeeffamu aerobic respiration jennani. Anaerobic means when cellular respiration is take place in the absence of oxygen, especially it is carried out in microorganisms. It is known as fermentations. When we see the difference between this aerobic respiration and anaerobic respirations, aerobic respiration is glucose is broken down into carbon dioxide and water molecules, then energy is released. Then this aerobic respiration is especially carried out in cytoplasm and the mitochondria. Bear in mind, aerobic respiration is carried out in presence of oxygens and also carried out in or occurs in cytoplasm and mitochondria. Cytoplasm and mitochondria. When we come to anaerobic respirations, it takes place in the absence of oxygens. Oxygen jirati gaggeeffama. Maal jedhama karaa biraatiin fermentation jedhama jechuudha. It is occurs in cytoplasm. It is occurs in cytoplasm. Isa keessatti gaggeeffama cytoplasm qofatti gaggeeffama jechuudha. But when aerobic respiration is, we come to aerobic respiration, it is carried out in cytoplasm and mitochondria. Aerobic respiration in isa keessatti gaggeeffama mitochondria fi cytoplasms. Much less efficient, producing only 2 ATP during glycolysis for every glucose molecule process. Baay'ee gad bu'aadha jechuudha efficiency isaa. Anaerobic respiration dhaan nuti ATP meeqa qofa oomishama 2 molecules of ATP is produced only, but in aerobic respirations we produce 34 ATP. So it's very important way of cellular respiration. The end product of anaerobic respiration is like ethanols and the lactic acids. Bu'aan inni dhumaa inni productiin anaerobic respiration ethanol fi carbon dioxide, lactic acid fermentations. Ethanol and the carbon dioxide in ethanol fermentation when used in beer and the bread making. Yeroo baay'ee yeastiin yeroo daabboo fi biiraa oomishnu in the producting ethanol fi carbon dioxide when yeast in beer and the bread making. But lactic acid fermentation is when our cell muscle during heavy exercise in animals in heavy exercise. So let me see this process in details. Glucose is converted into pyruvate by the process of glycolysis in cytoplasms. Then oxygen is present in cellular respiration when it is entered into mitochondria. But when the reaction is pyruvate is only in microorganisms, it produces ethanol and lactic. There is no oxygen is present in fermentations. So this is called anaerobic respirations. This reaction is called anaerobic respiration. Oxygen is absent, absent, but this is anaerobic respiration because oxygen is present in these respirations. When we comparing of aerobic respiration and anaerobic respiration, aerobic respiration if anaerobic respiration will be qabnee ilaallu takko takkotti ilaallumu one by one. This aerobic respiration occurs in cytoplasm and the mitochondria. Aerobic respiration kun kan gaggeeffamu mitochondria fi cytoplasmidha. When we come to this anaerobic respiration, it occurs only in cytoplasm. Eessa qofa keessatti gaggeeffama? Cytoplasm qofatti gaggeeffama jechuudha. This aerobic respiration requires oxygen. It is carried out in the presence of oxygen, but it does not require oxygen. Oxygen hin barbaadu jechuudha. This is more efficient. It produces 36 of 38 ATP, but this produces 2 ATP only. Baay'ee efficientidha. ATP 36 oomisha. Kun immoo baay'ee gad bu'aadha. ATP meeqa oomisha? 2 qofa oomisha jechuudha. You see the difference between this anaerobic respiration and aerobic respirations. So the students of grade 12 and grade 11s, please use these materials. It's very important for your understanding and also you answering the question related with this topic and use for national exam preparations. Good. For my students from general biology courses, please attend this course and also listen and then take a note, and also I hope you got good marks. And also the result in production of more waters and the carbon dioxide in aerobic respirations. The last in the product is waters and the carbon dioxide. Bishani fi carbon dioxide dhumarratti oomisha. When we come to anaerobic respirations, the end product is alcohol, carbon dioxide, lactic acid in lactate fermentation, and alcohol in alcoholic fermentations. Complete oxidation of glucose, but it is partial oxidation of glucose. Glucose oxidized godhuu hin xumura. Kun immoo partially oxidize the glucose. This is a difference between aerobic respiration and anaerobic respiration. When we start from seeing detail of aerobic cellular respiration, gadi fageenyaan aerobic cellular respiration haa ilaallu. In details, let me see about aerobic cellular respirations. These processes use glucose, which have 6 carbon molecules, Aq12O6 plus 6 oxygen molecules, which means the glucose is lost hydrogen atoms or by oxidations, and also oxygen is gain of hydrogen atoms by reduction reactions. Lastly, we get 6 glucose carbon dioxide molecules, 6 water molecules, and 36 or 38 ATP molecules. This is the overall reaction of anaerobic cellular respiration, the more efficient one. Inni kun reactionii aerobic respirationiti glukoosii fi oksijiinii walitti dhufanii yoo glukoosiin hydrogen atomii release godhu oksijiin immoo hydrogen atomii gain yeroo godhu molecuulii glukoosii carbon dioxide 6 molecuulii bishaanii 6 fi energy 36 hanga 38 argachuu dandeenya jechuudha. Kun overall reactionii kan aerobic cellular respiration. This is the overall reaction of aerobic cellular respirations. When a food or glucose is completely oxidized into carbon dioxide and water molecules and the energy in aerobic cellular respiration, then that stored or energy molecule is called ATP or adenosine triphosphates. So aerobic respiration is from metabolisms. It is a catabolic pathway or anabolisms. You remember about anabolisms and the catabolisms. Catabolisms or catabolic pathway means the process we break down large molecules into smaller ones, or break down carbohydrate into glucose, break down starch into or break down lipid into fatty acid and glycerols, or break down protein into amino acids. So in this process, we use oxygens. So oxygen is consumed as a reactant along with organic compounds. We consume oxygens as a reactant along with glucose. Aerobic cellular respiration is take place in three main stages. Have three stages. Aerobic cellular respiration gulantaa meeqa qaba? Sadii of keessaa qaba. It have three stages. So please, we see one by one these three stages, and please follow me. And like, subscribe my channels, and share for your friends, because I will present very smart and very good topics, and also by good manners. Channel kanaaf haaraa yoo taatan channel kana like, subscribe godhuu hin dagatinaa. Yeroo baay'ee namoonni emailii hin qabne subscribe waan isin hin gooneef emailii akka banattan isin hin gaafadha. Bifa bareedaa ta'een channel sagantaa bareedaa ta'u isin dhiyeessaa jira waan ta'eef, kan immoo isin na jajjabeessitan immoo channel kiyya yeroo subscribe gootan, yeroo namni baay'een daawwatuu fi channel groupii mana barnoota keessanii university keessanii irratti yeroo share gootan immoo baay'ee inspire na godha jechuudha. Let me see these three stages of aerobic cellular respiration. The first one is glycolysis. The first one is glycolysis. And also the second one is Krebs cycles. And also the third one is electron transport chains. Glycolysis means anaerobic respiration occurs in cytoplasm. Glycolysis in iddoo oksijiiniin jirutti fayyadama, gaggeeffama, ammallee cytoplasm keessatti gaggeeffama, carried out in cytoplasms. So the preparation for Krebs cycles, the must outside inside cytoplasms, pyruvate is converted into acetyl-CoA, which is past the membrane of mitochondria. The second one is Krebs cycles. This Krebs cycle is occur in matrix of mitochondria, matrix or outer membrane of mitochondria. Then after the Krebs cycle is over, the NAD and the FAT-H is carried out hydrogen atoms or protons into inner membranes or crystal of mitochondria by electron transport chains, which is carried out in inner membrane of mitochondria. These are the three stages of main stages of aerobic cellular respirations. Let me see one by one. Follow me. This is a diagram. It simply shows how or what is the stage of aerobic cellular respirations. The first one is glycolysis. This process is carried out in cytoplasms. Then it have a 10 steps. Gulantaa kudhan of keessaa qaba. 10 steps. Then glucose is converted into 2 3-carbon molecules called pyruvic acids by 10 steps. It is carried out in steps and they produce only one ATP. Only one ATP. Which means it produce one ATP means it produce 2 ATP, then it produce 4 ATP, and the net is 2 ATP, because when glucose is break down into the secondary molecules, they use ATP. Then this pyruvic acid is not enter into the membrane of mitochondria. Then it must convert into acetyl-CoA. Then it release carbon dioxide molecules. Then we come to Krebs cycles, which produce ATP and also carbon dioxides. In these Krebs cycles, we are also NADH and the FADH too. They enter into electron transport chain. Glycolysis, Krebs cycle, and electron transport chain, and the chemosmosis is the three stages of aerobic cellular respiration. Let me see one by one. Please follow me and also give me attentions. Bear in mind and take a note. If you have materials. So when we come to one by one in details, let me come to glycolysis. Glyco means glucose, lysis means a split or breakdowning. So splitting of what? Sugar molecules into two. The first step in degradation of glucose is glycolysis. It is the first step of degradation of glycolysis. Step jalqaba itti glucose caccabuu jalqabu maal jedhama? Glycolysis jedhama. Which break down glucose to pyruvates. It is breakdowning glucose into what? Pyruvates. Glucose have carbon-6 molecules, then pyruvate is carbon-3 molecules, converted into or breakdown into 2 pyruvate molecules. Partially oxidize glucose or carbon-6 molecules into pyruvate 3 carbon molecules. Occurs whether or not oxygen is present. If oxygen is not present, it does not matter. It's carried out in the absence of oxygen. This glycolysis is carried out in the absence of oxygen. This pathway is used by anaerobic as well as aerobic oxygens. This is a common stage for anaerobic organisms and the aerobic organisms. This process occurs in cytosol of a cell or cytoplasm of our cells. The processes take place in cytoplasm of prokaryotic and eukaryotic, but it does not require oxygen. The glycolysis does not require oxygen. The process is called exergenic process, which means energy is released. Most of energy is converted in high energy electron of NADH and in phosphate bond of ATP. So the process is called exergenic process, which means energy releasing process. The most common pathway for glucose degradation to pyruvate is found in animals and plant and microorganisms. It has 10 steps and also 10 enzymes. This is a review of glycolysis. We have glucose, 1, 2, 3, 4, 5, 6 carbon 6 molecules. Then we convert the process is converted into 2 pyruvic acid molecules. During this process, we have 2 ATP molecules are produced, 2 NADH is produced, 2 NADH is produced. The processes have 10 steps in biochemical pathways. The process of this glycolysis have 10 steps in pathways. Let me see the steps of glycolysis or the enzymatic reaction of glycolysis. This enzymatic reaction of glycolysis is called the enzyme, 10 enzymes, which is catalyze these glycolysis reactions. The phosphorylation of glucose at carbon 6 by hexokinase forming glucose 6 phosphates. Our cell is used hexokinase enzyme and the ATPs and forming glucose 6 phosphates. Glucose 6 phosphates. The first stage of glycolysis, G6P, glucose 6 phosphates. Okay, the second stage is conversion of glucose 6 phosphate to fructose 6 phosphate. Then conversion of this glucose 6 phosphates. So, by phosphorus isomerase enzymes convert or conversion of glucose 6 phosphate to fructose 6 phosphates by rearrangement enzymes isomerase phosphoexose isomerase. This is the second enzymes. The third one is phosphorylation of this fructose 6 phosphate to 1,6-bisphosphate by phosphofructokinase. Phosphorylation of this fructose 6 phosphates. Then the cleavage of this fructose 6 bisphosphate by aldolase enzyme, this yield two different products. Dihydroxyacetone phosphate and the glyceraldehyde 3 phosphates. By enzymes, aldolase enzymes. They produce two products. The first one is glyceraldehyde 3-phosphate and also dihydroxyacetone phosphates molecules. In fifth stages, isomerization of this dihydroxyacetone phosphate to second molecule of glyceraldehyde 3-phosphate by triose phosphate isomerase. Triose phosphate isomerase enzymes, the fifth enzymes. The sixth one, the six stages, in the six stages, the dehydrogenation and the concomitant phosphorylation of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase enzyme. This glyceraldehyde 3-phosphate dehydrogenase enzyme is converted glyceraldehyde 3-phosphates into 1,3-bisphosphoglycerate. Then, in 7 steps, the transfer of one phosphate group from 1,3-bisphosphoglycerate to ADP by phosphoglycerate kinase enzyme. Then, in the 7 stages, we have the ATP is produced, and 3-phosphoglycerate is produced. Then the isomerization of these 3-phosphoglycerates by 2-phosphoglycerate by phosphoglycerate mutase enzymes. And also this 2-phosphoglycerate is dehydration of 2-phosphoglycerate to form phosphoenol pyruvate by enolase enzymes. This is phosphoenol pyruvate by enolase enzymes. Lastly, the transfer of the phosphate group from phosphoenol pyruvate to ADP by pyruvate kinase. Then lastly, we get ATP and the pyruvates. We have ATP in 7th and 10th stages. Produce ATP. Then this stage is double stage. We have 4 stages in 4 ATP in glycolysis. This is the step of this glycolysis by one. This is glucose step 1. ATP and they produce glucose 6-phosphate. Then this glucose 6-phosphate is also converted into fructose 6-phosphate. This fructose 6-phosphate is converted into fructose 1,6-bisphosphate. 1,6-bisphosphate converted to glyceraldehyde 3-phosphates. This stages 1 up to 4 is energy investment stages or preparatory phases. The second phase up to 9 is energy payoff phases, which means energy is produced when 1,3-bisphosphoglycerolic acid or 2 molecules is converted into or produce ATP, and 3-phosphoglyceric acid molecules converted into 2-phosphoglyceric acids. Then phosphoenolic acid is converted into pyruvic acid and they produce this ATP. 1, 2. We are used to ATP, then we have double stage, 1, 2. Then we are finish this stage and then apply by 2. So we have 4 ATP, but we are using 2 ATP. So 4 minus 2 is equal to 2. So the net ATP of glycolysis is 2. The net ATP is 2. Well, when we summarizing this glycolysis summary, it is take place in cytoplasms. You remember this in shorts. It does not use oxygen, which means it is anaerobic respirations. It requires or input to ATP. It used to ATP. It glucose. It is split glucose into two pyruvate molecules. Also, it produce energy carriers to NADH for ATP. So lastly, glycolysis is produced for ATP, but net ATP is to ATP. Like glycolysis keessatti qaamni keenya NADH lama ATP afur oomisha. Garuu enerjiin yookaan net ATP yookaan ATP glycolysis arganne meeqa lama ta'a jechuudha. Lama maaliif duratti fayyadamne jirra waan ta'eef. So we are using 2 ATP in the first stage and then we minus 2, which means what we are investing in the first stages. We investing 2 ATP and negate 4 ATP, then our profit is 2 ATP. So the net ATP in glycolysis is 2 ATP. Good. Let me come to the second stages. It's called preparation for Krebs cycles or link reactions. Because this pyruvate is not enter into or pass the membrane of mitochondria or matrix. So it must convert it into another compounds. Intermediate process that convert pyruvate yookaan pyruvatein one cell membrane mitochondria seenuu dandeenyeef garamti jijjiiramuu qabaa gara acetyl-CoA jijjiiramuu qaba. So it must pyruvate is converted into acetyl-CoA which enters to TCA or citric acid cycles for complete oxidation of Krebs cycles and electron transport chains. Kanaafuu dirqama TCA seenuudhaaf membrane san keessa pass godhuudhaaf pyruvatein kun garamitti jijjiiramuu qaba. It must converted into acetyl-CoA. This pyruvate is transported to mitochondria. Garamitti transported ta'uu qaba. It is transported to mitochondria where it is decarboxylated into acetyl-CoA. Gara seenuudhaaf dirqama maal ta'uu qaba? Gara acetyl-CoA jijjiiramuu qaba jechuudha. By pyruvate dehydrogenase enzymes. By pyruvate dehydrogenase enzymes. This pyruvate dehydrogenase enzyme is the enzyme which converts pyruvate into acetyl-CoA. You bear in your mind like this. Sammuu keessanitti kan argatu. Enzymiin pyruvate gara acetyl-CoA tti jijjiiramu pyruvate dehydrogenase enzyme jedhama jechuudha. So this process is the junction between glycolysis and the Krebs cycles. The process where pyruvate is converted acetyl-CoA is called link reaction between glycolysis and the Krebs cycles. The enzyme complex or pyruvate dehydrogenase converted pyruvate into acetyl-CoA by the following chemical changes. Once decarboxylation of pyruvate is loss of carbon dioxide. The linkage of acetyl group to coenzyme A, then making 2 carbon compounds forming acetyl-CoA. Pyruvate is lose carbon dioxide, then it is acetyl group is group to coenzyme A, then making 2 compound molecules, then for lastly form 2 compound or acetyl-CoA molecules. Simply this is a reaction of link reactions. We have pyruvate, carbon 3 molecules, then this carbon 1 is converted into these carbon dioxides. Then we have NADs get hydrogens, it become NAD H and 1 hydrogens. Then it produces acetyl-CoA, 2 carbon molecules, acetyl coenzymes A. So this acetyl-CoA is enter to mitochondria, Krebs cycles stages. So during these times, 2 NADH is produced per glucose. 2 NADH is produced per glucose. In previous class, we have for a NAD in until pyruvates. The second stage is Krebs cycles or citric acid cycles, TCA cycles. So Krebs cycle means a Krebs cycle is considered as central pathway of aerobic respiration or metabolism. It is found between glycolysis and electron transport chains. These Krebs cycles have two purposes. The first one is bioenergetics, which means resulting in release of energy and high energy molecules or electrons. It have two importance: release of energy and high energy electrons, which means NAD, H, and FAD-H. The second one is biosynthesisi, which means it supplies precursor for several biosynthesis pathways of amino acids and the purines. It have precursor for several biosynthesis pathways of amino acid and the purines. This process requires energy. Krebs cycle requires energies. It is complete breakdown of glucose, but glycolysis is partial breakdown of glucose. It is take place in matrix of mitochondria. Matrix of mitochondria. The H2 or hydrogen atom is produced here is not gas, but it bound to co-substrates called NAD, H, and FAD-H, which is subsequently oxidized in respiratory chains. It's passed to inner mitochondria called crystals. So this Krebs cycle is a cyclical series of oxidation reactions that give off carbon dioxide, ATP, NADH, and the FADH is the end product of Krebs cycles. We have the four end product of Krebs cycles. The first one is carbon dioxide. The second one is ATP. The third one is NADH. The fourth one is FADH2. This is the end product of Krebs cycles. End product of Krebs cycles. The first step of Krebs cycle is each acetyl-CoA, which means two carbon molecules bonded to oxaloacetate or four carbon molecules. Acetyl-CoA is two carbon molecules. It's bonded with oxaloacetate, four carbon molecules. Then they form sister citrate or carbon-6 molecules. The first stage of Krebs cycle is acetyl-CoA is bind with oxaloacetate, four carbon molecules. Then it forms new molecules called citrate or carbon-6 molecules. This is acetyl-CoA, carbon-2 molecules. This is oxaloacetate, carbon-4 molecules. They form citrate, carbon-6 molecules. This citrate undergoes 5-step cycles that build additional ATP, NAD, and FADHs in these Krebs cycles. Citrate is eventually back to oxaloacetate. This citrate and the cycle is continue in another steps. The Krebs cycle is turned twice per glucose molecules because you have two pyruvate, which means two acetyl-CoA. So the cycle is turned twice. Kanaafuu staging Krebs cycle kun turn twice. Yeroo lama deebi'a jechuudha. Because pyruvate lama waan qabnuuf acetyl-CoA lamaatu uumamaa. Kanaafuu yeroo lama jechuudha. ATP tokko argannu lama arganna. Afurii argannu 8 argannaa jechuudha. Well, this is the stage of Krebs cycles in diagram with a beautiful diagrams. Diagramii baay'ee bareedaadhaan kan isin dhiyaate. We have acetyl-CoA, two carbon molecules. Then this acetyl-CoA is bind with oxaloacetate, 4 carbon molecules. This is 4 carbon molecules. This is 2 carbon molecules. They form 6 carbon molecules called citric acids or citrates. Citric acid or citrates. Then this citric acid molecule or citrate is converted into isocitrate, 5 carbon molecules. Then it releases carbon dioxide and also NADH. This isocitrate is also converted into alpha ketoglutarate, which means carbon 4 molecules. It's also release glucose and the NADH. And also these 4 carbon molecules converted into another 4 carbon molecules. Another 4 carbon molecules. It may be malates. Malate. It's converted into another 4 carbon molecules. Malate. Then it forms or produce ATP. These 4 carbon molecules converted into another 4 carbon molecules. That's called fumarate. Called fumarates. So during these 4 carbon compound is converted to another 4 carbon compounds, it is release FAD H2. Then these 4 carbon molecules also change into new another oxaloacetate molecules, which is continue these Krebs cycles. Then it release also NADH. So we have 1, 2, 3 NAD, which means when times 2, because the cycle is 2, 6 NAD, 2 FAD H2, then 4 carbon dioxides. Let me see by different stages. So in step 1, acetyl-CoA enters mitochondria and combine with oxaloacetate to form what? Citrates. This acetyl-CoA is combined with oxaloacetate by enzyme citrate synthetase enzymes. It forms citrates, carbon 6 molecules. This citrate is converted into isocitrates by aconitase enzymes. This forms isocitrates. This isocitrate is converted into alpha ketoglutarate. It produces carbon dioxide, NADH and also carbon dioxide by isocitrate dehydrogenase enzymes. This alpha ketoglutarate is changed into succinyl-CoA by and forming NADH and carbon dioxides. This carbon dioxide is succinyl-CoA is converted into succinates. Succinates. You see, by succinyl-CoA synthetase enzymes. Then it produces ATP, when succinyl-CoA is converted into succinate. This succinate is also converted into fumarate when it produces FADH. Then fumarate is converted into malate and release water molecules by fumarase enzymes. When malate is converted into oxaloacetate, it also produces NADHs. Let me count the amount of NAD and FADHs. Citrate is further oxidized, produce 3 NADH and FADH2. 1, 2, 3, 4. 1. Let me count this row. 1, 1, 2, 3, 4. 3 NADH and 1 FADH2. And also 2 carbon dioxide are produced as citrate is converted back into oxaloacetate. Let me count the carbon dioxides. 1 and also 2 carbon dioxides. When we times 2, we have 4 carbon dioxides. And also oxaloacetate is ready for another acetyl-CoA and the Krebs cycle continues. Because once it is reacted with, then the reaction is also produce another oxaloacetate and also 1 ATP. Generally, in the Krebs cycles, cycle is carried out complex degradation or oxidation of acetyl groups or 2 carbon molecules unit in acetyl-CoA. It produces carbon dioxide, resulting in release of energy ATP and also reduced power of NADH and FADH. It produces carbon dioxide, energies, NADH and FADHs. which means results 2 carbon dioxides, 1 ATP, 3 NADH, and 1 FADH per turnus, 1 turnus. We have 2 turnus, turnii lama qabna jechuudha. Don't forget, we have 2 turnus. The cycle turns twice. You see, the cycle turns twice per glucose molecules. Therefore, for each glucose molecules, the Krebs cycle is produce 4 carbon dioxides, 6 NADHs, 2 FADH, and 2 ATP. 2 FADH and 6 NADH, 2 FADH and 2 ATP. Because the turn is twice, the cycle is double. In third, the cycle produce only 2 ATP, but load up coenzyme with hydrogen atoms and electrons, which move to the third stage of electron transport, cellular aerobic respiration called electron transport chain and chemosmosis. This is overall of Krebs cycles. The third one is, or the third stage of aerobic respiration is electron transport chains. This is the third stage of aerobic respirations. This electron transport chain is collection of molecules embedded in inner membrane of mitochondria or crystal. It's carried out in inner membrane of mitochondria. This electron transport chain is a point at which most of ATP is produced by oxidative phosphorylations. Electron transport chain kan gaggeeffamu inner membrane of mitochondria keessatti gaggeeffama. It is ATP kan oomishamuuf most of ATP is produced by electron transport chain oxidative phosphorylation oomishama jechuudha. Elektiroonii yookaan pirootiinii naadichi irraa baadhatee dhufu. Oxidative phosphorylation in this stage means the overall process of ATP synthesis is associated with flow of electron from what? NAD, H and FADH. Oxidative phosphorylation jechuun ATP as keessatti oomishamu hundi isaatu elektiroonii naadichii fi FADH irraan darbamu kan oomishamudha jechuudha. In the presence of oxygen, bakka oxygen argamutti. Dhumarratti final electron acceptor eenyu ta'a? Oxygen ta'a. It produce water molecules. Water molecules. It produce water molecules. So the two components of oxidative phosphorylation in electron transport chains. We have two components of electron transport chains in this electron transport chain. The first one is electron transport chain and the second one is chemosmosis. Let me see one by one. Oxidative phosphorylation step lama qaba. Electron transport chain fi electron transport fi chemosmosis kan jedhamu qaba jechuudha. Let me see one by one. Electron transport means in these stages the electron from NADH and the FADH is passed through electron transport chains. The electron from NADH and the FADH to has passed electron transport chains. There is a chain in mitochondria, then the electron from this NADH and the FADH hydrogen or proton carrier is passed through electron transport chain. The flow of these energies or electron along these pathways cause protein complexes to pump hydrogen from mitochondrial matrix into inner membranes. When this electron is flow from matrix to inner mitochondria, the hydrogen is pumped into inner membranes. Then it creates proton concentration gradient. The proton concentration in the inner membrane is become high. Then the more energy is formed, or ATP synthesis converted this proton to ATP. So the storing energy as proton is motive force or hydrogen gradient. Storing energy in this proton is called what? Motive force or hydrogen gradients. Energy proton yookaan hydrogen keessatti stored kun maal jedhama? Motive force jedhama yookaan hydrogen gradients. The second stage is chemosmosis. This chemosmosis, after this motive force or hydrogen gradient is enter into the inner membrane, production of ATP using energy of hydrogen gradients across membrane to phosphorylated ATP. By using these hydrogen gradients, It is phosphorylated ADP and converted into ATP. Phosphorylated ATP jechuun ATP ADP dha. Phosphorus ADP yeroo itti dhiyaatu ATP ta'a jechuudha. As hydrogen is diffused back into matrix through ATP synthesis, it powers ATP synthesis. Waan hydrogeniin gizee gara maatriksii bahu ADP'n gara ATP'tti uumamaa jechuudha. Drives phosphorylation of ADP to ATP. ADP'n gara ATP'tti kan jijjiiramu godha jechuudha. High energy electron move along electron transporting and lose energy in several energy releasing steps. High energy electron warra jennuun kun warra kan ADH fi FADH jechuudha. At the end of the chain of aerobic respiration, electron is passed to high oxygen. It is reduced to water molecules. So the final electron acceptor or terminal electron acceptor in this electron transport chain is oxygen, then it produces water molecules. So the process of oxidative phosphorylation is produce a maximum of 34 ATP or 90% of glucose. Electron transport chain is produce 34 ATP or 90% of glucose. So for every 5H molecules, 2 ATPs produced. One 5H is produced to 2 ATP, but one NAD is produced 3 ATP. One NAD is equal with 3 ATP, and also one FAD is equal with 2 ATP produced. Therefore, from one glucose molecules, we produce 34 ATP molecules generated in electron transport chain by oxidative phosphorylation alone. We produce 34 ATP by oxidative phosphorylation. Kanaafuu electron transport chain keessatti ATP 34 oomishamaa jechuudha. Adeemsa oxidative phosphorylation kan jennu jechuudha. Yookaan elektirooniin baadhatamee in the form of ADH fi FADH deemu sun isa kan oomishaa jechuudha. Let me work over all of these stages. We have two NADHs from glycolysis, which means it produces 6 ATP, and also two NADH by formation of acetyl-CoA, it produces 6 ATP. We have 18 ATP because 6 NADH is formed in citric acid cycles, which means one NADH is equal with 3 ATP, 3 times 6, 18. We have 2 FADH in citric acid, which means 2 times 2, 4. We have 34 ATPs produced by electron transport chain. I hope it is clear for you. It's very clear for you. So this is a stage of electron transport chain. The stage of electron transport chain. Intermediate space, inner mitochondrial membrane, and the mitochondrial matrix. This is outer mitochondria, this is inner mitochondria. We have electron flow, NADH, then NADH. When this release, one hydrogen protein hydrogen is pumped out from here. Then electron is passed from one complex to second complex. Then the electron from this NADH is come here, and the final electron acceptor is oxygen. Final electron acceptor is oxygen. It produces water molecules. Then these hydrogens pass here and they come to enter and it produce ATP by ATP synthesis enzyme. So this stage before here is electron transport chain. This stage, then when a hydrogen is converted to ATP is called chemosmosis. The ATP synthesis is powered by the flow of hydrogen back across membranes. It's called chemosmosis. This process is generally called oxidative phosphorylation. Oxidative phosphorylation. So this is generally the overall reaction of aerobic respirations. We have 3 stages: glycolysis, Krebs cycles, and the electron transport chain. In glycolysis, we have 2 ATP produced in glycolysis, and also there is no ATP in acetyl-CoA or link reactions. In Krebs cycles, we have also 2 ATP. These 4 ATP is produced by substrate level phosphorylations. When we come to electron transport chain, we have 34 ATP, which is produced by oxidative phosphorylation. So the maximum ATP which produced in the cell is 38 ATP. This is the overall energy of, or ATP of, aerobic respiration is 38. This glycolysis is carried out in cytoplasmic fluid. This electron transport chain and the Krebs cycle is carried out in mitochondria. This overall reaction of so an account of ATP production by cellular respirations. Let me see one by one. Glycolysis. In glycolysis, we have 2 ATP by substrate level phosphorylations, 2 NADH, which produce 2 NADH means it produces 6 ATP by oxidative phosphorylations. The amount of ATP depends on how this NADH is propagated to 80 electron transport chains. Then 1 NADH means to produce only 2 ATP sometimes. When we come to formation of acetyl-CoA, there is 2 NADH, which means 6 ATP. 1 NADH means 3 ATP, and also 2 ATP. 6 ATP is produced by formation of acetyl-CoA. In Krebs cycles, we have 2 ATP by substrate level phosphorylation, 6 NADH, 18 ATP. 6 means 1 NADH is equal with 3 ATP. So 3 times 6, 18 ATP by oxidative phosphorylations. 2 FADH2 is produced 4 ATP by oxidative phosphorylations. Let me count. 6, 18, 6, 24, this is 4, 28, 2, 13, 6, 36, 2 ATP, 38 generally. So grand total sometimes 36 to 38 ATP per glucose. One cell is produce this glucose ATP per glucose. When this the passage of this NADH is instead of this there is 2 NADH fat and we get 36. Sometimes it becomes 38. Overall reaction of cellular respirations. This notes. It's a very specific good notes. Depending on how NADH is produced in glycolysis, gets to etc. The maximum ATP generated from one glucose molecule is 36 or 38. Sometimes 2 NADH is produced in glycolysis make 4 ATP. Sometimes the glycolysis, the NADH that is produced in glycolysis is converted into 4 ATP. Due to this reason, maybe we get 36 or 38. In brain and skeleton, the muscle of human and in bacteria, the maximum yield 38 ATP per glucose. But in our livers, in heart and the kidney cell of human, maximum yield 36 ATP. From our cells, the cell which produce 36 ATP is liver, heart and the kidney produce 36 ATP. Qaama keenya keessaa onnee keenya, tiruu keenyaa fi kaleen keenya ATP 36 qofa oomishachuu danda'u. Garuu sammuu keenyaa fi iskeleetanii keenya keessatti immoo ATP 38 oomishamaa jiru. And also in bacteria 38 ATP is produced. 38 ATP is produced. This is the difference between in our cells. Seelii keenya keessatti in the brain and the skeletal muscles, our brain and the skeletal muscles produce 30 ATP, but our liver, heart, and the kidney cell of humans produce 36 ATP per glucose molecules. This is how ATP is produced in our bodies. This is glucose to pyruvate in glycolysis. We have 2 ATP, very smart diagrams. So we have 2 NADH, which means 6 ATP. In pyruvate oxidation, we have 6 NADH, 6 ATP, 2, 6, 6 by chemosmosis. When we come to Krebs cycles, we have 6 NAD, which means 18 ATP, and 2 FADH, 4 ATP. Let me count. 2, 6, 8, 6, 14, 18, which is produced by Krebs cycles. 18 plus 18, 30. 18 plus 18, sorry, 8, 6, 14, 16, 18, 34, 38. The total net yield of ATP in eukaryotic is 36 or 38. Especially 38 is produced in our brain and skeleton, but 36 is produced in liver, kidney, and heart. This is overall about aerobic respiration or cellular respiration. I hope you understand very well. It's very clear for you because I will show in different diagrams by different detailed illustrations. So I hope you understand it very well, and also you get a lot of things from these materials. Thank you for watching my channels. Thank you for watching the Double Education channels. Thank you for becoming a member of Double Educations. Baga miseensa chaanaalii keenyaa taatanii baay'ee isin jaalannaa. yuutuubii kanarraa yookaan viidiyoo kanarraa waan baay'ee akka fayyadamtan abdii guddaan isa qaba. Mamii tokko illee hin qabu because it's prepared very well. Baay'eetti qophaa'ee wanta isin hidhaate waan ta'eef waan hedduu irraa qabatan. Ispeeshaalii barattoonni kutaa 11 kutaa 12 qormaata biyyoolessaatiif deemtan wanta kanarraa akka fayyadamtan abdii irraa qaba. Thank you for watching Double Education. Please subscribe channel and share for your friends. See you next class. Bye bye.